Warmly welcome to today's seminar. Today we'll have the great pleasure of listening to Sohini Ramachandran, who will talk about the causes and consequences of human genomic variation. Sohini Ramachandran was awarded her PhD in 2007 at Stanford University's Department of Biological Sciences. She joined the faculty of Brown University in July 2010 after spending 3 years as a junior fellow at the Harvard Society of Fellows and as a postdoctoral fellow at the Department of Organismic and Evolutionary Biology at Harvard University. She is now Associate Professor of Ecology and Evolutionary Biology and Director for the Center for Computational Molecular Biology at Brown University. And we are very happy to have her here for this semester. Sohini's research engages with the very research frontier in her field and covers a range of significant questions such as what loci are under strong adaptive selection in the human genome? Are there genetic pathways we can identify that underlie common diseases such as diabetes? And are patterns of variation in human genomes similar to those of cultural traits, like language? Research in her lab addresses problems in population genetics and evolutionary theory, generally using humans as a study system, so to speak. The lab uses mathematical modeling, applied statistical methods, and computer simulations to make inferences from genetic data. And Sohini has, with colleagues, published significant works on these very issues. Her research has also been recognized and funded by the National Science Foundation, US National Institutes of Health, the Pew Charitable Trusts, and the Alfred P. Sloan Foundation. And here at Scas, Sohini is pursuing two new and very exciting research directions. The first is integrating ancient DNA and modern genomes into the interference of human population history. And this research goes on via collaboration with the Evolutionary Biology Center. And the second stream of research concerns developing a novel statistical framework for multi-ethnic disease mapping studies. And it is with great curiosity that I now leave the floor to Sohini.
Thank you very much, Christina, for that really kind introduction, and to all of you for taking time out of your afternoon to be here. I'm very grateful. And I'd like to start in particular with acknowledging the Swedish Collegium for Advanced Study and the Natural Sciences Program for giving me the opportunity to be here, both to pursue the research in my program that I've built back home, but also to strengthen my ties to the wonderful Human Evolution group here at Uppsala University, and also for supporting having my family here. Additionally, the history department has welcomed my husband, Jeremy Mumford, and so this is really a once-in-a-lifetime memory for all of us, which I really appreciate. So part of what drew me to Scas is that I have always sought out and really thrived in interdisciplinary environments, and that's because it may not be obvious to those of you who are not human geneticists, but human genetics is actually an extremely multidisciplinary field. It really relies on other fields to cast its results into relief. In particular, and this is really showcased by the work at the EBC here at Uppsala University, research on human evolution really relies on bringing together results from archaeology, linguistics, and genetics, and you could say that the project of the field of human evolutionary biology is to describe the history of our species over the last, say, 100 to 100,000 years. And I think the importance of this interdisciplinary conversation, which now extends to sociology, economics, demography, and historical anthropology, is best described in the book Genes, Peoples, and Languages by Luca Cavalli-Sforza. Sadly, we lost Luca in the past couple years, but he was a close collaborator of mine during graduate school and an invaluable mentor. So he says in the introduction to this book, really beautifully, that these three disciplines, archaeology, genetics, and linguistics, can be expected to converge towards a common story, and that behind them must lie a single history. To some, I quote from his beautiful intro, history, including evolution, is not a science, because its results cannot be replicated and thus cannot be tested by the experimental method. But studying the same phenomenon from many disciplines has the value of largely independent repetition. This makes the multidisciplinary approach indispensable. So I want to start here because in this talk, I'm actually not going to draw so much on other fields that are enamored with the human species, but rather on bringing quantitative approaches from other disciplines to bear on genetic data. But at the end of the talk, we'll arrive back at this statement, which has shaped my career, but I also begin here because I think this sentiment is what draws people to human evolutionary biology, the chance to understand the story of our species. So I have a very ambitious outline for the talk today, and I hope you're all caffeinated. So first I'm going to begin with a primer on population genetics, because this is a general audience, and also on human genomic variation. I know at Scas we like to be very genteel and save our questions for the end, but I invite you to ask me for clarifications, especially in this part of the talk. I come from a very interruptive field, so I don't want to lose anyone right at the beginning. In the second part, I'll talk to you largely about work from my increasingly distant past by myself and others, but also some recent work in my group on the signature of population dynamics or histories on the human genome today. Then in the third part, I'm going to talk to you about a project that is in review now that I finished after arriving at Scas in collaboration with one of my graduate students and others that for me is really overturning some of the things I thought we understood about genetic variation and is enabled by large-scale analyses of large biobank data sets. And lastly, I'll pose to you some of the challenges that genomics research faces in our current political climate that I would very much like the feedback of my Scas colleagues and others on. Okay, so to begin, population genetics is the study of the evolutionary forces that produce and maintain genetic diversity within a species. This is a biological population, the biological use of the word population. These forces are mutation and recombination in sexually reproducing species, migration or movement between populations, Darwinian natural selection, and random genetic drift, or the stochastic process within a finite population by which some individuals reproduce and some individuals don't. The terms that I've highlighted in green are forces that increase genetic variation or supply de novo genetic variation to a group, and the ones in red reduce genetic variation. So in order to make predictions about the future of a population or in order to infer the past, population geneticists develop models and simulations or analytical theory to make these predictions or inferences and compare them to observed data. Sometimes these are forward in time models, dynamical systems that rely on diffusion theory, and sometimes these are backward in time statistical models. So the goal of these quantitative approaches and of research programs like mine is really to generate testable hypotheses regarding the evolution of the species of interest, and then to validate those either with studies from other investigators or through new experimental collaborations. Okay, so that's my field in one slide. Now, the human genome. The human genome, which lies in all of our cells and all of our bodies, is 3 billion DNA base pairs long. It is aggregated into 23 pairs of chromosomes, which for our purposes today are just blocks of DNA that live in our cells. On average, any two of us in this room differ, though, only in 1 in 1,000 of these base pairs on average. We are quite similar genetically, despite differences we may see in the traits, the way we look, and other aspects of our health, say. the way that mutation happens is well approximated by a single mutation at a single base pair, a point mutation. There are other kinds of mutations that can happen, occurs at a rate of about 10 to the minus 8 per site per generation. So what this means is that between a child and their biological parents, there are probably between 30 odd to 100 new mutations observed in that child that are not observed in the parents. Some of those occurred during embryogenesis, and some of them occurred during the process of producing eggs and sperm in the parents before the embryo begins developing. So a lot of population genetics is predicated on the idea that this mutational process is well known and well modeled. And the idea of inference is based largely on a molecular clock. The idea that if we look at sites in the genome, the same sites across many individuals, the number of differences between individuals is correlated to the time since they had a common ancestor. So I'm going to illustrate that for you now. Suppose that we sample 3 individuals, and this is the present day, and let's say that these individuals and their families identify as, for multiple generations, being from Tibet, being Han Chinese, and being Danish. And let's say the DNA letters that we observe are A in these 2 Tibetan and Han individuals, and T in the Danish individual. So this is one site at the genome, 3 people. If we had to, based on our knowledge of the history of peopling of different parts of the world, just had to guess at a model to describe how these 3 individuals are related, we would probably draw a tree that looked like this. So going back into the past, we would say that the Tibetan and Han individual have a common ancestor more recently in the past than either does with the Danish individual. And then if we wanted to understand how this particular observed data came about, we could look at the DNA letter at this site in some kind of outgroup or in a primate genome. And let's say there that we observe a T. So this is our observed data. But there are many different ways that this could have come about with different mutations, and they each have different probabilities, different likelihoods. For example, this common ancestor to these 2 individuals could have had a T at this position in the genome, and 2 independent mutations to A could have happened after the divergence of these 2 populations. But this is actually quite unlikely. So there are lots of things that could have happened, but usually what we want to figure out is statistically, what's the most parsimonious explanation? So the highest probability explanation in this case is that some individual ancestral to these 2 individuals had a mutation from T to A, and then that was inherited here. So a lot of what happens in population genetics is doing this kind of comparison across many sites in order to understand these genealogical relationships, which we can't truly observe. And this is possible because our genomes are historical texts of a certain type. They are mosaics of our ancestors' genomes, and they offer insight into ancestral events. So to dive into this a little bit further, here are 4 people many of you have met. This is me and my husband and our 2 biological daughters. So if we got genetic data from all 4 of us, then we could think about my daughter's chromosomes. For every chromosome in their genome, they've inherited 1 from their father and 1 from me. But with enough genetic data, with our genetic data from their parents, and also with reference data from people who have similar ancestry to me and people who have similar ancestry to my husband, we could actually identify that they each have 1 chromosome of European ancestry at every chromosome in the genome, and 1 chromosome of South Asian ancestry. So more interestingly, if they go on to have their own children one day, they will pass on mixed ancestry chromosomes to their children. And those chromosomes will have dependent ancestry segments that I'm going to illustrate for the next couple of slides. So this happens because of the process of recombination, which is a mystery unto itself in cell biology and in genetics, but it's thought that in the process of proper cell division, chromosomes have to meet their mate, their pair, within the cell, and in order to properly separate, they have to connect and have a recombination event where genetic material gets shuffled. And approximately in the generation of eggs in a female or sperm in a male, there will be one of these meiotic crosses, breakpoints, for every chromosome in a proper cell division event. So the chromosomes that my daughters pass on one day, if they have their own children, might look like this. They might look 50% European, 50% South Asian, or slightly more South Asian ancestry, slightly less, and so on. These recombination breakpoints can occur at different parts in the chromosome, although they might be concentrated in certain parts of the chromosome. So this means that with large reference data sets, scientists can produce a chromosome painting, which displays DNA segments from different ancestries. So let's take a look at what one of these might look like. This is a product that is given to people as part of direct-to-consumer testing nowadays, and a product a lot of people enjoy looking at if you haven't looked at one yet. So this is from the blog of the direct-to-consumer testing company 23andMe in the US. This is one of their employees. Her name is Sarah. And her family history, the oral history, is that one grandparent is from Germany and met another grandparent who is from Ireland. They argue a lot about who makes the best beer. And then she has two grandparents on the other side of her family who are Ashkenazi Jewish. And she herself identifies as being Jewish. So this is what using their reference data set 23andMe does for her chromosome painting. I don't know how well you can see the colors in the back, but if you were to look closely, as close as I am, you could see that every chromosome, this is 1 to 22 and then X, she has two X chromosomes because she's female, there's one dark green chromosome, and that represents her Ashkenazi Jewish side of the family. Okay, so there are sort of three colors that are appearing representing her different ancestries in this chromosome painting. Let's contrast this with Antonio, who is from Mexico, who also works at 23andMe, and he identifies as Latino. His chromosome painting looks quite different. And the ancestry segments that he has in his genome correlate with Native American, African, and European ancestry. So the reason why I'm spending time on this, what's interesting about this is that the length of each colored chromosomal segment here is inversely proportional to how long it's been since the mixing event occurred. So my daughters have one European chromosome and one South Asian chromosome. You can see Sarah here in this 2 generation ago mixing event has these long chromosomal segments of the same color. But Antonio's ancestors began mixing much longer ago. And in fact, his genome bears signatures of their history, which is embedded with colonization and slavery and European conquest in the New World. So under the hood of how this chromosome painting happens is a machine learning technique that actually everyone in this room is very familiar with, which is hidden Markov models. And I know you're familiar with this because you've seen this at work if you've used your smartphone or Google email anytime recently. Because this is the same model that underlies predictive text functions, right? So, for example, in Google Smart Compose, or when you're writing a text, based on learning probabilities of certain phrases you write when a word is observed, you'll see that your computer, your phone, tells you usually the next 3 words that you might write. Sometimes you can write a totally grammatically accurate message this way, not always. But the chromosome painting problem is exactly the same. So basically you're saying, at this site in the genome, based on everything I'm observing from these labeled references, based on their ancestry, what's the ancestry I should assign? And you have a large penalty for changing ancestries. You need a lot of evidence to say the previous assignment is no longer the correct assignment. Okay, so it's the same kind of process. So what I'm going to tell you about today are machine learning frameworks that we create in my lab to study processes in human genetics and human evolution. But I also want to remind everyone that the question I'm really interested in is not just a computational question, but a biological one, which is, what are the relative roles of natural selection versus population histories in generating and maintaining human genetic diversity? So this is a figure from a review about how human studies of disease and genetics don't actually assay the diversity of our species. So these are individuals whose ancestries are usually left out of these studies. But when we think about our species, we have a very diverse phenotype. We've colonized and inhabited all kinds of ecosystems, adapted to different diets, faced different pathogens. What allowed us to do that genetically? Is the variation, that small level of variation amongst any two of us, how is it structured? Is it structured really based on population histories, or did it develop as an adaptive response to the different selection pressures our ancestors experienced? So now I'll tell you about some background work that I've done in this area, and other people have done as well. So a lot of the work in human evolution has benefited from sequencing technologies enabling the creation of large, publicly available databases of genetic variation. And one of the first ones of these, which Cavalli-Sforza pioneered the creation of, is the Human Genome Diversity Panel. I know it's hard to see this, but it's just a map with crosses. But this represents genetic data from 1,048 individuals, and there are 52 crosses, which represent 52 geographic locations these individuals were drawn from. They were drawn to be 2-generation indigenous to that part of the world. So when I started in this field as a graduate student, my research in the Human Genome Diversity Panel revealed a distinct genomic signature among human populations. And I'm going to explain this plot in detail now. So on the y-axis is a measure of within population genetic variability. So here you're seeing 52 points, one for each of those crosses on the previous map. And on the x-axis is geographic distance of that population to Addis Ababa, Ethiopia, a putative origin for the human expansion out of Africa. But the paths are computed to not cross oceans. So the way that the paths were computed, say, to get from Africa to the Americas were to go over the Bering Strait, the way that our ancestors did. So the takeaway here is that geographic distance computed using these coarse waypoints for geographic paths really predicts within population genetic diversity. So what kind of model could give rise to this pattern? The model that we proposed, and others had proposed as well around that time, is called the serial founder effect model. And I'll describe it to you in a cartoon now. So suppose we have a population, and the colored boxes I'm showing you here represent genetic diversity in this population. And say the population now is resource limited, and some people decide to move, and they take their genes with them, and they found a new population, and that population grows, and then this process, let's say it just repeats itself. This is a really simplistic model, and the major force in it is these founder effects, or what are sometimes called bottlenecks. This is really, what's shaping this model is random genetic drift. What's happening in the course of this cartoon is that you can see at every step we lose some colors, we lose some genetic diversity. In fact, the diversity we observe as we move down the chain is a proper subset of what we observe earlier in the chain. Okay. And so you could think about this as a process where there's migration into a new continental region crossing a barrier, and there's not a lot of migration backwards. So de novo evolutionary variation, things like mutation rates, are too small to counteract these founder events. And in fact, the predictions of this simple model turn out to be very robust in modern human genetic variation. So contemporary genomes observed today. As an example, this is just looking at the number of different, you could say, sequences at different locations in the genome that we had access to in 2005. And again, the same pattern is recapitulated. So this is different from what I showed you on the previous slide, but another prediction that this model would make. After that initial work, through collaboration, we got access to samples in the Americas, which were not initially studied broadly in the Human Genome Diversity Panel. So this is Canadian First Nation groups and Meso and South American Indigenous Americans. And it turns out that the same pattern is borne out. So the within population genetic diversity in Indigenous American groups is less than that outside of the Americas. And there's also a higher variance within population genetic diversity, which in a follow-up project with a colleague, Noah Rosenberg, we described as representing a rapid coastal peopling of the Americas, followed by a very slow inland migration. And we also controlled for the variable effects of European colonization on these particular sampled groups. Again, with different technologies, this pattern continues to be observed robustly in contemporary human genomes here using genome-wide, what's called SNP chip data. And there's another very interesting prediction of the serial founder effect that I'll spend some time on later today. And that is an increase in the length of shared sequences with distance from Africa. And this is actually work by my colleague Matthias Jakobsen, who's here and who's at EBC and Uppsala University. So what this is showing is that recombination has had more time to break up associations between shared sequences in African populations, whereas as you move away from Africa, there's an increase in the shared genetic variation that occurs within populations. So there's less genetic variability as you move away from Africa. So this is the same pattern, but sort of observed in a different way. Okay, so there's a strong signature of population histories on genomic data. So why do we care? Well, one reason we might care is that modeling this process is going to be crucial to understanding any mutations that might be disease-causing or adaptive in the human genome. And identifying, for example, mutations that allowed our ancestors to survive as they faced new ecosystems, new pathogens, new diets, is a crucial part of human genetics. So I'm going to tell you a little bit about recent work in my group along these lines. So one common model of adaptive evolution is called a sweep model, and I'll illustrate this for you with a cartoon. So here, each of these lines is a genomic sequence, and the little balls are different variable mutations you might see across people. You can think of each of these as a person's genome. And this star is a beneficial mutation. This person gets it randomly, and it allows them to survive and reproduce differentially from others. So over time, this star will increase in frequency until it's fixed in the population. Everybody has it. It's really adaptive. Not only will that happen, but anything that is in that person's genome, let's say, as a first approximation, will also increase in frequency. These non-selected neutral alleles will get dragged to a high frequency, and this is called the hitchhiking effect in genomics. I call these mutations the friends of someone who becomes really famous on reality TV. They just go along for the ride. Okay, so how can we, in the face of this kind of rapid evolution, use computation and statistics to localize an adaptive mutation with all this genomic data that we have now? So this is a project that's led by a wonderful postdoc in my group who's about to start her own faculty career this summer, named Lauren Sugden. And what she did was leverage different statistical signatures of adaptive evolution. So these are population differentiation. Maybe there's a mutation that's adaptive for these people on this side of the mountain, not for these people. These long shared sequences, and then other changes to the distribution of allele frequencies, for those of you in the room who are biologists. And we're not the only people who have done this. A lot of people want to do this. But our goal was to create a really interpretable approach to do this and to localize the adaptive mutation. Okay, so what does this have to do with what I've spent the last 20 minutes telling you about? Well, the issue is that neutral population dynamics and adaptive evolution can leave similar signatures on genomes. So how do we differentiate between these? I'm going to show you simulated data from just a neutral population history, and then the same history with an adaptive sweep. And I'll show you summaries of the data in a two-dimensional space. So these are contour plots, so these likelihood surfaces are sort of coming out towards you. So these are the sweep simulations that have the same history as these neutral simulations. This is a statistic measuring population differentiation, FST, for those of you who know. And this is a measure of long shared sequence data, or IHS. Okay, so what I want you to think about is, if I were to sort of squish down to just this marginal distribution here, if I try to draw a vertical line to separate the sweep simulations from the neutral simulations using just this axis, I can't really do it very well. Nor can I draw a horizontal line and do it here quite well. Neither of these statistics on their own can differentiate between these two evolutionary scenarios. But in this space together jointly, I could draw a boundary and maybe classify these well. Okay, so the machine learning problem is to draw on multiple statistics and do what's called supervised classification. So this is like when you tag yourself in a photo on Facebook or in your Google Photos, you're training a computer to then ask you when you upload a new photo, is this you? Or is this your friend that you tagged in your photos before? This is the same machinery here. Can we learn what happens to the genome under these different scenarios and then try to predict something from data? So it turns out we can. And we find actually that modeling these population histories is crucial to identifying adaptive mutations, and we can do that using genomes alone. So here I'm showing you genomic position on the x-axis. And this is in African individuals, and this is around a gene called DARK, or the Duffy antigen receptor for chemokines. It's known that this gene contains mutations that confer resistance to malaria, which is really important for people living in this part of the world. And our algorithm is called Swiffer, which might be funny for Americans in the audience. It detects sweeps. And so on the y-axis is the probability our algorithm gives. It gives a probability that there is an adaptive mutation at this site. Are there Swiffers in Sweden too? No, okay. All right. Okay, so this is 50% probability. So no adaptive signal, something small, but oh look, this mutation has a 99% probability according to Swiffer of being adaptive. And it turns out that this is a variant that has been known to confer resistance to Plasmodium vivax. And this was discovered in the 80s and 90s using traditional molecular techniques. So what we're trying to propose with this approach is that we can provide very targeted hypotheses for experimental validation of adaptive mutations. And this is one validation that might be the case, but one goal is to reduce false positives and also to give a very targeted site, saying, I think this is the causal allele for a functional assay. We then applied this genome-wide to SNP, CHIP, and exome data, for those of you who are biologists in the audience, collected by Brenna Hen's group in the Khomani San. So this is another group that studies African genomics extensively. Again, one of the major strengths of the UU Human Evolution group is the wonderful work they do in African genetics. When we scan genome-wide for adaptive evolution signals in the Khomani San, we find that the Swiffer signals are enriched in genes associated with metabolic and obesity-related phenotypes. So I'll show you what this looks like. So these are the chromosomes. This is every position in the genome we had data at. And these are the Swiffer probabilities. There's a pink line at 50%. And I've highlighted genes that had were enriched for high probability Swiffer signals of sweeps. So these genes turn out to be annotated using molecular experiments as influencing body mass index, triglycerides, predisposition to diabetes, and also the generation of adiponectin, which is secreted only in adipose tissue and the human placenta. So we think this is really interesting, in part because it's believed that our ancestors who lived as hunter-gatherers were likely able to retain fat readily because they had to for times of famine. And that fat is also crucial to successful gestation in times of famine. And so we think that we may be seeing a signal of adaptation across the genome that might be related to these traits. And in particular, in this gene ADIPO-Q, we observe a high-frequency variant in these Khomani San individuals that across many public databases we only see 2 copies of this allele in there. So we're really interested in learning more about that. Okay, so what have I just told you in this kind of first part or second part? How do population dynamics influence observed human genetic variation? There's a strong signature of our origin in Africa in extant human genomes. The human expansion out of Africa and migrations since associated with, for example, the development of agriculture in multiple continents are characterized by these founder events and little cross-continental back migration, at least when you look at contemporary genomes. And also modeling the signature of human evolutionary history is crucial for understanding how natural selection has shaped our genomes. Okay, so now for something completely different. I'm going to talk to you about homozygosity, and I'll define that. But now we're looking within a genome, and homozygosity is inheriting the same DNA letters from your mother and father at many positions in the genome. So this is thought to be very deleterious because it can unmask deleterious mutations. And a classic picture in high school biology textbooks in the US is this one. This is of an Amish child from Lancaster County with Ellis Van Creveld syndrome. This is a syndrome which causes a child, and that child grows up to have very short stature, but also polydactyly, so more than 5 fingers or toes on each hand, each leg. This child has 6 fingers on each hand. Due to the founding of the Amish community in the 1700s in the US from a very small set of families, it turns out that this deleterious mutation reached very high rates in the Amish because of their endogamous marriage practices and was traced back to one couple that lived in Lancaster County in 1744. But it turns out that genome-wide runs of homozygosity within a genome in populations are not rare, and in fact, they reflect the predictions of the serial founder effect model. This is not my work, but this is work by a colleague in my field, Trevor Pemberton. And again, here populations are ordered by sort of distance from Africa. And here, what's being shown in these little plots, these are called violin plots, is a depiction of the distribution of total runs of homozygosity in these populations. So for those of you who aren't familiar with these plots, what I want you to take away is that these are the African numbers. These are pretty small, but they increase with distance from Africa, just like we might expect. So this is sort of related to that signature of long shared sequences that I showed you before. But it turns out that very long runs of homozygosity, as long as 1 million base pairs in the genome, and remember, we only have 3 billion base pairs in our genomes, these are really long, in fact, can be found in groups that practice endogamous marriage. So here what I want you to look at is these long tails that occur in really the Middle East, Central South Asia, maybe to some extent Oceania, but also in indigenous American groups. Okay, so for example, in South Asian caste groups, like my ancestors, very long runs of homozygosity are observed. And these are largely thought to be not a good thing. So I wanted to understand how runs of homozygosity might be distributed within a single human genome. Now I'm going to tell you about work that's a collaboration with 23andMe. The paper is online, which I just say to say that the data are not, well, the results are not private. But this was led by a graduate student of mine who just defended in September. So just to tell you about the 23andMe database, which is extremely enormous, but the analysis of these large biobanks of data that merge medical records along with genetic data is becoming more and more common in my field. So this data set has over 5 million people in it who've given their saliva to 23andMe. 80% of them consent to being part of research, and through surveys, they've released over 1 billion different data points about their traits in aggregate. So initially, this is the question we were asking, but then this was a data science project, and we ended up pivoting to a new question because parent-child data at 23andMe revealed a peculiar genomic signature between a biological parent and child. So what 23andMe, one of the things they do if you opt in, is they'll show you genomic segments that are shared between you and other individuals in the database. And in fact, customers find relatives this way. Many adult adoptees have found their biological family in this way. But here's a Reddit post a customer shared of her results with her parent, one of her parents. So this is what the output in the product looks like. So these are multiple chromosomes, 11 to 15. This individual, this is her comparison with one of her biological parents, and she's half identical to this parent on all these chromosomes. And that's what we would expect. We get half our genome from each parent. But then on chromosome 16, there's another pattern, this gray, which the legend says means this individual has no genetic relationship, is not identical at all to this parent on just this chromosome. So how can this happen? We typically learn that, I've told you, we get half our genome from our mother and half from our biological father. So now I'm going to tell you about individuals for whom this is not the case. And these individuals have uniparental disomy. So this is the inheritance of an entire chromosome, an entire pair of chromosomes, or a large segment thereof from just one parent, with no representative copy from another. So how does this happen? Well, in the process of cell division and the formation of eggs and sperm, we start out, and I realize these might be kind of small for those of you in the back, but we start out with what are called sister chromatids, then there's a shuffling of genetic material with recombination, a splitting, and eventually the formation of 4 daughter cells, one of which goes on to fuse with another cell and produce a proper, what's called disomic zygote. So this is what happens normally. But the way that uniparental disomy can happen is if there's an error early on in this process, you can end up with, instead of having sort of half a genome in eggs or in sperm, a sort of full genome, pairs of chromosomes. And many things kind of have to align, but you can basically end up with this situation where on one or multiple chromosomes, you only have genetic representation from one side, from one parent. This is thought to be very deleterious generally, and that's because it's been studied clinically. So sometimes children present in a clinic, they have sort of strange symptoms, and they and their parents are genotyped, and it's identified that they have UPD. And the first clinical case was documented in 1987. It's still common to have case reports that report a handful of cases, even just one case observed in a clinic. Since then, over 3,300 clinical cases have been reported. This is from a database online, the life's work of a physician. And this is the chromosome-wide distribution of observed cases. There's been a case observed on every single chromosome. There are cases where the genetic material that's left is inherited from the father, cases where it's inherited from the mother. So from this data set, maternal UPD is 1.5 times more common than paternal. Many cases are observed on chromosomes known to cause what are called imprinting disorders, medical diseases that happen when one parent's genetic material is overexpressed versus another. But there are many open questions. The one open question is, what is the incidence of this in the general population? Are all cases of UPD, do they all result in a medical diagnosis? What are the rates of different types of UPD? How often is it that the maternal genetic material is what survives in the offspring versus paternal, say? And what's the etiology of UPD? Okay. So this is a very rare phenomenon, and you really need data-driven science to sort of characterize it in this way. All right, so how can we do that using computational approaches? Well, it turns out, just bear with me a bit longer, and then I think more things will make sense. There are 3 subtypes of UPD. In one, called heterodisomy, you inherit both copies of one parent's chromosome. So you inherit, instead of material from all 4 of your grandparents, you inherit material from just 2. But this coloring is meant to show you that there's going to be genetic variability in this individual. It's just going to come from only one side of their family. In another extreme case, you can have what's called isodisomy. You can just get one copy of one chromosome identical from one parent. So here, no variability, full homozygosity on millions of base pairs. Or you can have something in between, which is called partial isodisomy. Okay, so if we have parent-child genetic data, we can detect all of these quite easily. Without that, though, you need to look at these runs of homozygosity. And in this case, you expect to see no large blocks of homozygosity, but in these 2 cases, you do. Okay, so first we looked at parent-child genetic data. So this we're able to sort of solve exactly. And across 1 million parent-child pairs, approximately, in the database, we identify 199 individuals with UPD, none of whom had a diagnosis, none of whom volunteered that they had UPD. This is the per chromosomal distribution. We find that the overall rate is 1 in 2,000 in the 23andMe database, which is rare, but actually not that rare. And existing estimates were much lower. So we find this is the first estimate of the rate of UPD in the general population, and it's twice as big as past estimates. We also find that maternal origin UPD is much more common than paternal origin UPD. And we see that the clinical descriptions of UPD are very biased towards chromosomes on which this event causes a medical disease. So here I'm showing you our per chromosome rates from the 23andMe database on the y-axis, and the x-axis is the clinical published data that's available in this database online. So the takeaway here is just that there is no statistical correlation between these two sets of data. So the clinical studies are biased towards one set of chromosomes and miss other observations. Okay, but if we don't have parental data, can we identify candidates of UPD just using runs of homozygosity? So this is a computational problem. I'm going to show you what the machine learning problem looks like. So first I'll show you what do runs of homozygosity look like in a normal individual, no UPD. Okay, so this is going to be impossible to see, but later on perhaps not. So here this individual in green has a few long runs of homozygosity, about 3, 4 here, that you can barely see, that are green. And here's an individual who actually has heterodisomy of chromosome 1, of the biggest chromosome, but you don't see any green there either, right? Because they basically got the full complement of genetic variation on that chromosome from just one parent. But now here's someone who has partial iso-UPD. So a little bit of material from both parents, but mostly from one parent. You see these long green blocks. And here's someone with full isodisomy. Okay, so this is hopefully easy to see in the back. So these are different. And this is the computational problem. How do you train a model to distinguish these two pictures from these two pictures? Why is this difficult? It's difficult because now suppose I show you what UPD of chromosome 1 looks like in an individual who has parents that are related. Let's say distantly related, but related because of inbreeding at a large scale in the population. So now, this is what these individuals truly have UPD of chromosome 1, but you can't quite tell because there are other events down here. You might classify as UPD, and for this individual, you might say they have UPD in chromosome 9, but not in 1, because you can't identify that heterodisomy event. But to further complicate things, an individual without UPD in this population also is going to have these long runs of homozygosity. So how do you train the model? So it turns out this is quite hard to do in some populations, Ashkenazi, Jewish, and South Asian being 2 of them. It's easier on long chromosomes than on short chromosomes. But we were able to gain power to do this, and we found an additional 304 cases out of over 1 million people in the 23andMe database. And we were able to replicate this in another data set. So we can't sort of figure out the parent of origin, but this is the distribution. We observe an event on every chromosome. We also see a high correlation in this data and what I showed you from the parent-child data are true positives. So how does UPD arise? Well, it's a way to solve an error in cell division. And we know that errors in cell division in the formation of eggs and sperm happen. They happen with age, particularly in mothers. So it turns out that in our data set, mothers who have children with UPD are on average older than mothers who have children without UPD at the time of the child's birth. And we couldn't replicate this in fathers. And this might make sense because previous studies show that other errors in cell division and miscarriage rates increase with maternal age. Okay. But interestingly, we didn't really find strong phenotype associations with UPD. Now, there are some issues with trying to do this. So basically what we're trying to do here is say, let's take our individuals with UPD and the traits they volunteer that they have, are they enriched for certain traits that other individuals without UPD are not? And this is not an ideal experiment to deal with self-reported data, but you could also argue that someone who's put their genetic data in a database is kind of interested in the genetic underpinnings of their traits. So we found some kind of statistical association with lower weight and shorter height and having UPD of chromosome 6. But the strongest association we found, which maybe Terry has some thoughts on, is of UPD of chromosome 22 and being on the autism spectrum. Some marginally high effect size, and quite significant even when correcting for the number of phenotypes that we were testing for. Okay, so it's been long believed that homozygosity is quite deleterious, especially across long stretches of DNA. But we know that populations have different levels of homozygosity based on population histories, but we still don't really understand what maintains homozygosity within individuals. And UPD has never been characterized comprehensively using computational algorithms until this study, but we estimate it's much more common than thought in live-born individuals. And interestingly, and I didn't phrase this in the most scientifically rigorous way, but I think what this points to is actually that there's very strong embryonic selection against individuals in whom UPD unmasks deleterious mutations. And actually the individuals who are born with it don't seem to have ill effects. So there's more to this story and to understanding how errors in cell division can be fixed. Okay, so then to kind of arrive at some provocative concluding remarks, what I'm hoping I showed you is that the challenge in human genetics today is really one of methods and theory, and how do we understand the data that is washing over us in modern human genetics. To give you a sense, if you're not in the field, of what this looks like, here is the NIH's statistics on the cost of sequencing a human genome over time. This is 2001 to 2015, and this is a log scale from $100 million to 1,000. Okay. So just to cast my age into relief, this is when I started graduate school, this is when I started my postdoc, this is when I started my job, and this is when I got tenure. Okay, so tough crowd, okay. A lot has happened in my adult life in this field, and really genomics isn't data limited in many ways, although of course there are parts of the world we're not comprehensively characterizing the genetic variation of, but it's really limited by the ability to make sense of the data. So what happens when we try to make sense of the data from human genomes? Well, genetics can just underscore how complicated human dynamics and the mixing of human population is. It's often accompanied by aggression. So here I'm showing you a somewhat old analysis now of US self-identified African Americans and their genomic data. On average, US African Americans have about 80% genome-wide African ancestry and about 20% genome-wide European ancestry. Okay. So if you move across chromosome 1 and do this chromosome painting, you're going to see variations in individuals in these numbers. So some individuals will have 78% African ancestry, some will have 40% African ancestry. But overall, in this particular analysis, no one deviated much from this median estimate of about 78%. This is chromosome 1. The X chromosome, which men inherit from their mothers, had a different story, which is that in the female X chromosome that was inherited by individuals was overwhelmingly of African ancestry, underscoring that in the past of these individuals, their genetic ancestors were meeting in a sex-biased way, really European men raping African women during the context of slavery. Similarly, studies of genomic ancestry can continue to shed light on the complexities of self-identification. This is another analysis of African American and European American self-identified individuals. Okay, so what I'm going to show you here is this is the self-identification This is the proportion of genomic African ancestry, from 0 to 2% in the first bin to 98 to 100% in the last bin. This is the proportion of individuals in each bin. So I'll uncover this slowly so we can break it down. For individuals who genomically have between 0 and about 25% African ancestry in the US, most of them identify as European American. Some identify as African American, but most of them identify as European American. Once you get to about 25% genomic ancestry, which can correspond to about one African ancestry grandparent, this changes completely. So there's still outliers. There's still individuals who have a lot of African genomic ancestry who might identify as European American and vice versa. But here you can see that for mixed ancestry individuals, the way that they identify and the way society classifies them is quite complex. Genomic technologies have led to more accurate prenatal diagnosis, but with societal consequences. So last week, March 21st, was World Down Syndrome Day. Here is a picture from a beautiful article by Amy Harmon, who's one of my favorite New York Times science writers. This is from a series she did in 2007 that won a Pulitzer Prize called The DNA Age. It's a beautiful series of articles. And this one talks about Down syndrome. In the US, about 5,500 children are born each year with Down syndrome. But now, today, 90% of pregnant women in the US who are told that their fetus has Down syndrome will choose to have an abortion. And what that means, what she talks about in this article, is that the state is putting less money towards research on Down syndrome, even though individuals with Down syndrome have an average life expectancy of 49 years. There's less educational infrastructure for people with Down syndrome in the US and less institutional support in general. Genomic information can also give us agency at our doctor's office, but at least the American medical system is really ill-equipped to handle this. So this is the cover of Time magazine from 2013. Angelina Jolie, the actress, had a prophylactic double mastectomy at that time. Her mother died of breast cancer, and she found out using DNA testing that although genetically influenced breast cancer is extremely rare, she had multiple mutations that meant she had about an 87% chance of having breast cancer. And she had the prophylactic procedure and has not had this illness in that intervening time. But actually, in response to this, direct-to-consumer testing companies in the US were told to cease and desist giving this kind of information to consumers, in part because it was thought that an individual shouldn't get this information without a medical doctor explaining it to them, even though medical doctors are not really trained in genetics, at least in America, nor in statistics. I shouldn't say it so biasedly, but anyway, this is a problem. And lastly, increasingly, genetic studies are misinterpreted and used as fuel for sexist or heteronormative or white supremacist movements. This is a protest, or a Trump victory party that happened in New York after he was elected. And what you see here are men with their shirts off, showing that they are white by chugging milk, which is something that is pretty rare in most parts of the world, but quite common for European Americans. So most of the world cannot digest milk as adults, but Europeans can. And this is actually a kind of neo-Nazi salute to whiteness, the ability to drink milk and aggressively chug milk in these kinds of ritualistic gatherings. So, what's a geneticist to do? I think that our field has really tried to avoid these kind of fringe, misinterpretations, but it's a problem now. And in fact, unfortunately, just a couple of weeks ago, the Christchurch shooter, in his horrific manifesto, posed the following question, I'm quoting, and then answered it. He wrote, "What makes you believe that there are racial differences and that those differences matter? Research and data, haplogroups, phenotypes, and globalized testing. In time, the truth will be revealed." So I don't know where we go from here, but I think it's time for geneticists to not be silent about the context of our work, about how similar we all really are, and to sort of step back and wonder the extent to which our work can easily be used to reinscribe the idea that there are differences that are meaningful amongst us, which I don't really think is the case, at least certainly not when it comes to people's self-worth or value to society. So I'll pick it up a bit by noting that the heavy lifting is all done by my wonderful lab, who's made it possible for me to be away. The work I told you about today was done by Lauren, this wonderful postdoc who's now fledging the nest, and Priya, who defended, and also a young graduate student named Sam Smith. And we are grateful to our funding agencies and collaborators. And thank you very much.